So thank you very much, Mark, for the kind invitation, and it is a great honor for me and a great pleasure for me to be here and to be one in a distinguished row of speakers. I looked at the internet whom I succeeding, and I must say I'm very proud and pleased that I can follow on this series and give you this lecture in memory of Peter Gallagher. I do not even try to make any jokes or any statements about rugby play. No, what was it? Yeah. I hardly know the name, and I hardly know what soccer is. And you know, soccer in Germany is a very popular type of sports. So I completely restrain from doing any statements on that. But really thank you that I can be here and then that I can deliver this lecture tonight. And I hope I can convince you that sugars are the right structures to address if it comes to finding specific targets also in tumor biology. And I hope and I think Peter Gallagher, who has supported this institute significantly, as I learned from Mark, would appreciate our research because, as I learned also, he died sadly from cancer. So I hope this type of lecture would have pleased him. Okay, with that said, I start and introduce to you what I called sweet tooth viruses for cancer treatment. So I have to introduce a few different topics to you, and I hope you won't mind that from time to time I have to be a little bit scientific. But otherwise, I try to make it understandable also to the public. So let's start. So I give you first of all a structure of my talk. I put a glance on glycobiology or on biology to explain the value of sweet teas in tumor targeting. Then I will explain some strange words like oncofetal antigen and molecular mimicry. Both are mechanisms that are used by cancer cells to escape our immune surveillance system. Then I will introduce the term oncolytic viruses or viral induced tumor lysis or oncolysis, acquaint you a little bit with viruses that eat bacteria, and those viruses we call bacteriophages. Then describe how we used a combination of bacteriophages and phages that infect humans, and we stole from the bacteriophages the sweet teas in order to transplant them onto the human phages, the human viruses, to make them these sweet tooth viruses. So let's start with what we all love the most here in this Institute for Glycomics and also in Hanover in the cellular chemistry. This is the sugar shell of the cell. Each cell from outside is sugar, and this is true for all cells that are living organelles. And these sugars are not simply flattening or lying on the cell's surface, but the sugars are bound to proteins, as illustrated here and here, or they are bound to lipids. So they are not just candies lying on the surface of the cell. And the bulk of sugars that we have on our surface, the bulk of the glycan structures that we expose, is terminated by a specific sugar, which is, again, the most beloved subject in the Institute for Glycomics and also in my institute. And the name of this sugar is sialic acid. And just to honor a little bit also our setting here in our the beautiful Institute of Glycomics, I show you just one chemical structure, and this is the structure of sialic acid. Afterwards, sialic acid will only be illustrated by this diamond here, by this violet diamond. So in this outstanding position, in this exposed position, you can imagine that the sialoglycoconjugates, or the sialic acid per se, is involved in a number of cellular interactions. In cell-cell recognitions, like for self or foreign, it is involved in almost 100% in infectious disease or viral or bacterial infection. So many infectious diseases intruders first make contact to the outermost sugars, which is sialic acid. There are toxins that can bind to sialoglycoconjugates, and of course viruses and bacteria. And another situation that comes on the cell surface and is created by sialic acids can make long chains, so-called polymers. And if they do so, they are growing to a large volume, and this is illustrated with this sphere. This is because this sugar can bind an enormous amount of water, and that makes the carrier molecule a very large structure. So, and what is it doing in this situation? It regulates cellular contacts. Here we have a situation where two cells, these ovals here are cells, and these are the surface structures in the absence of polysialic acid. And you can see the cells can come close to each other and can make very stable contacts via these different molecules. If polySia is present, these structures or these contacts can no more be formed. And polySia increases the distance between cells. It weakens the interactions between these cell surface structures, and it gives cells more freedom to move. So it increases the motility of cells. And this is very important, as we will see later on. When do we have polysialic acid in our body? We express it naturally. And when we look into mouse tissue with an antibody that specifically recognizes polysialic acid, and we look at tissue homogenates from different days after birth, this is 1 day after birth, 2, and so on and so forth, what we see is that polySia is highly expressed in the young tissue and then gradually decreases and, importantly, is virtually absent in the adult. There's almost no polysialic acid left. And this is very important in the context of our today's lecture. So what we saw that was in the middle of the 90s of the last century, we thought we should know what happens if we knock out polysialic acid. And this is what we did. We used genetic technologies that are all easily available, and these are standard technologies, meanwhile, to create a mouse in which polysialic acid has gone. We call this a knockout mouse. So, and what we now have is a mouse that has no polysialic acid anymore, and as I said, we call this mouse a polySia knockout mouse. So how does it look in real life? This is the polySia knockout mouse at postnatal day 28. So this is after birth, day 28, and this is the littermate. And as you can see, this is the control mice, so the healthy littermates. They develop normally, they grow and give a normal mouse. However, these animals that cannot produce polysialic acid anymore, they do not thrive. They are very skinny and they are very ill. And as you can see from this mark here, they die before they reach the age of 4 weeks. So, and what is the reason for dying in these mice? Actually, we don't know. We have a few reasons identified, but the majority of reasons is still unknown. But they die because of a multi-organ failure. So, and this would tell us all organs, basically, for proper development, need polysialic acid. And I show you, again, just one phenotypic component because it is fitting very well with the context of our today's talk. And this is the fact that neuronal cells lose their capacity to migrate. So this is a particular tract in the brain, and this is a wild-type situation where we stained the cells that make this tract. And as you can see, this is nicely formed in the wild-type littermates. And when we look into our knockout animals, the polySia knockouts, this tract is virtually completely gone. And when we stain this structure, so you see it's stunted, it's not developing. So what is missing is these cells cannot grow anymore. They cannot extend. And that fits with what we discussed before. polySia conveys the potential to migrate, to be motile. And this is missing in these cases here. So what have we learned so far from these experiments? So we learned that polysialic acid is necessary for animal development. If we knock out polysialic acid, we have an early postnatal death because of a multi-organ failure. polySia is, however, almost completely absent from the adult healthy tissue. And lack of polysialic acid, as I showed you in the last slide, obviously interferes with cellular motility. However, polySia also in the adult reappears, and it reappears in the situation of a tumor. So tumor tissue, and in particular tumors that grow with high malignant potential, so very aggressive tumors, have lots of polysialic acids on their outside. And polySia as such behaves as an oncofetal antigen. I will explain this term a little later. So it appears early in development, in the adult has disappeared, but comes back in the situation of oncologic development, and this is in the tumor. And this is what we call an oncofetal antigen. Oncofetal antigens are very important for the tumor because the body, our immune system, is acquainted to these structures. So it is not recognized as foreign. So having an oncofetal antigen means the tumor cell can cover itself with a well-known antigen, and is not seen by the immune system. So this is an important mechanism of the tumor cell to protect itself against immune attack. So, and here I summarize once more what polySia does in the tumor and how it augments metastatic growth of tumors. So here again we have a situation a little bit more natural where two cell surfaces come close to each other, stabilization takes place via surface structures, and if these surface structures are polycellulated, as shown in this cartoon, and then they move apart, each cell gains motility, each cell gains more chances to migrate and find new places. And this is not healthy for the body. So it destabilizes cellular contacts and improves motility. It, however, also protects the tumor cells against the immune system. And both components are very important for the tumor to find new places to settle down and regrow and make metastasis. So virtually what we can say is a tumor cell is a smart cell. It uses a protective shell, and it comes as a wolf in the sheep's skin. And this is the way it undermines our immune system. So results and consequences from this knowledge are polySia is an oncofetal antigen. I explained the term already. It is a structure that is transiently expressed during development and disappears in the adult, reappears in cancer cells. The consequence from this is polySia provides a tumor marker because in the adult we have a polySia negative environment and only the tumor is polySia positive. And thus it is a target to find, to search for tumor cells and to destroy tumor cells, as we learn later on. The challenge for this ambitious goal is we need good reagents to chase behind the polySia tumors, the tumor cells. And this is where the bacteriophages now come into play. So the bacteriophages, I introduce them here. Well, I go one little step back. So polySia, as a colleague from Greece called it, the nature's ultimate stealth system. It can make invisible structures that are underlying because it is so big, and even structures that would be down here would be covered by this shell. And we have now learned that tumor cells are using this mechanism to escape the immune surveillance system, but there are other bugs or other guys that also do the same, and these are meningococci. So we have neuroinvasive bacteria that cover themselves with this polySia shell to undermine our immune system. And this is a mechanism that we call molecular mimicry. So the bacteria are using completely different enzymes to make the same polySia shell. So they make the same structure with using another principle, and this is why we call this molecular mimicry, and thereby blind the host immune system. However, the bacteria, in contrast to our immune cells, to our tumor cells, are not safe in this closing of polysialic acid, because there are viruses which eat bacteria, and this is bacteriophages, and there are bacteriophages that have polysialic acid degrading activities. So they have these tail structures, and some of these phages have a tail structure that is capable to bind and degrade polysialic acid. So then they dig these holes into the polySia capsule, and what they then do is, so they bind, screw their tails down to the bottom or to the membrane, and then inject their genetic material. And after they have done so, they assemble inside the bacterial cell. And after they are done, they lyse the bacterial cell to gain access to the outside world and access to new bacteria and restart a new infection cycle. So I have been interested in these enzymes or in these bacteriophages for many, many years, almost 30 years. I have been studying these phages. And because of their selectivity and high specificity for the degradation of polysialic acid. And here you can see the bacteriophage, how it really looks. This is the reconstruction, and a molecular reconstruction of the virus or of the bacteriophage. And here you can see these tail spike proteins that specifically degrade polysialic acid, and this is a blown up of the structure which we are now looking at and which we call endosialidase. So again, results and consequences. Bacteria are not safe in their polySia closing. This is different from the cancer cells. The cancer cells are safe, and this is because in our own body we do not have any systems like endosialidases that would degrade polysialic acid. So what can we do? We thought, as a consequence, we should make such a system. We should create a system which allows the detection of polysialic acid by, for instance, viral particles, and then use these viral particles to kill these cells. And as a basis to start with, we used adenoviruses. Here is an adenovirus. This is, viruses have many shapes and many structures. I will not go into any details of this. But an adenovirus is not a particularly dangerous virus. It is mostly infects children, small children. It causes symptoms that virtually can go from head to toe. Most of the symptoms are respiratory and intestinal symptoms. The attractive thing in this adenoviruses is that they are for many years studied as oncolytic viruses. So I tell you a little bit about the entry mechanism of these viruses. So this green structure here is very important for the entry into the host cell, in the mammalian cell. And at the end of this green structure, we have the so-called fiber knob. And this fiber knob binds to a specific receptor without giving any name. But this receptor is essential for the first contact between adenovirus and mammalian cell. This is a type of tethering. They go to this receptor, they are held, and then come closer to the membrane, and then many other proteins are involved in taking up the virus. So a virus-induced lysis of cells has the following steps. So the virus goes inside the cell by the mechanism I just explained. Then it starts replicating, it lyses the cells, and it releases its progeny to the outside world, which then can induce a new infection cycle. In this cycle, which is not much different from what we learned from the bacteriophages before when they infect the bacterial cells, the problem, however, in this system is that the first contact which is made between these fibers and the surface of the mammalian cell involves this CAR receptor, this important protein. And this protein is downregulated in the tumor cell. So the tumors do not express many of these proteins. However, the healthy cells have lots of this CAR receptor. And that means if you do this type of infection, it is mostly the healthy cells that would be affected and would die by the lysis of, by adenoviral-induced lysis. So there must be a protection for the healthy cells. You cannot use the virus as such to lyse tumors. There must be a stop sign that prevents infection of normal cells. And yes, people have invested a lot of energy and a lot of time to engineer these viruses in a way that they cannot amplify in normal cells. So the virus still can infect, infection takes place, but it cannot amplify. So we have no progeny. And this is very important because the viruses can amplify in cancer cells. They can infect and also amplify in cancer cells. Still, this first infection step takes place also in the normal cells. And you can imagine that this causes a massive cytotoxicity also for healthy cells that don't need to be lysed by the viruses, rather should be protected to be not affected by the virus. So our goal then was targeting polySia positive tumors, because as we learned, there are many tumor types that re-express this structure. And because we have this nice phage protein, the endosalidase, we thought we should combine this phage protein with the adenoviral strategy of tumor lysis in order to bring the virus specifically into the tumor cells. So the plan then was, we are doing a chimeric virus with the fiber knob replaced by an endosalidase. We did so. This is the virus again, and what we have done is we added the endosalidase to the end of this green fiber so that the fiber knob was covered and replaced. And exactly, roughly 4 years later, we had this virus in hand, and we also gave it a name, and this is the name that we gave to the virus. So this is, I now made a big jump because making this virus was not really easy. And to convince you that we really have this virus that we were looking for, I show you 2 electromicrographs. And this is the wild-type virus, and I hope you can appreciate that this virus has these long structures with the small fiber knobs. In contrast, our new chimeric virus, as it is shown here, is having much shorter fiber structures and is having these globular heads, which are the endosalidases that I showed in red before. So once more, the principle of our concept, and I appreciate for the fact that now the colors have changed. What was red before is now bluish. Sorry for that, but I couldn't change the color. So this is a scheme of the adenovirus, of the chimeric adenovirus, with the endosalidase at the end of the fibers. When we use this virus, it can no more bind to the CAR receptor. So the natural receptor is no more a receptor for the adenovirus. However, we have a new receptor structure, and this is polysialic acid. So the virus now migrates to cells that have polysialic acid exposed on their surface. And then, because this is an active enzyme, this is a degradase, this enzyme cuts down the polysialic acid, brings the viral particle close to the membrane, and then the other mechanisms that I mentioned already before, starting and are endocytosing this virus. The question, is this all true, what I told you? This is nice theory, but is it really working in praxis? Yes, it does. So this is the first example, made in vitro. So we used cell cultures that are positive for polysialic acid. And we cultured these cells in the presence of decreasing concentrations of the viruses. We used this chimeric virus that I explained to you, and we also used the wild-type virus. And when you see these roundish structures, these roundish cells, these are dead cells. And if you look at the very last panel here, so you see large holes in the loan of the cell culture loan. So these are massively reduced numbers of cells that are left in this well here compared to this one here, which is full of cells. So what does this tell us? This tells us this virus is taken up very well by polySia positive cells, and it is very capable to lyse these polySia positive cells. This one, in contrast, cannot very well infect these polySia covered cells, and therefore the cells can start initially with high doses. We see cytotoxic effects, of course, but then they dilute out very fast, and you see with the lowest dose there is no effect at all. So this is in vitro study, but we always do careful in vitro studies before we start doing animal models. So, but we have done an animal model in this case also. So we used our chimeric virus and asked the question, can we treat tumors in a living system? And the first model you normally go for in this type of studies is an immunosuppressed mouse. So what we did here, we used tumor cells, human tumor cells, which are highly positive, then injected these tumor cells in the flank of the animals, and then waited until, so we gave them time, about 5 or 6 days, until the tumors had accumulated, the tumor cells had accumulated in the flank and were growing to a tumor mass, as it is shown here. You can easily see the tumor. And then we started our treatment, and we did 3 groups. So the first group was our chimeric virus. The second group was the control virus, with the original adenovirus from which we started. And then a third group is a saline control, so where we only injected sodium chloride into the tumor mass. This injection is done exactly directly into the tumor. Yeah, and these injections into the tumor mass were done every second day. So and now we look at the results. So this is a time point when all tumors, sorry, that was too fast. This is day number 5 after transplant injection. So then you see the xenograft tumor has formed in this size about. And then at this day, we started our injection. So you must see the tumor, otherwise you couldn't inject into the tumor mass. This is really important. So this is the first injection. We did several injections. I grow them up a little bit faster. So, and this is the saline injection. And you see after only 13 days, the mice had disseminated tumors. So the tumor mass was high, and we had to kill the animals because you cannot go on if the tumor reaches a certain size. But by the size of the error bar, you can appreciate that these mice had massive tumors with very different size. And they were really ill. Then we did, with the parental virus, we saw a little reduction. So you can see this virus, which can only amplify in the tumor cells, was pretty effective, or at least compared to the mock control. We had a significant effect in survival of mice and reduction of tumor mass. With our chimera, we saw virtually no tumor growth. We saw a complete reduction of the tumor, and so saw virtually no recovery of the tumor. At the very end, we saw a little going up, but at that day we had to kill the animals. We could not go on, so that I cannot tell you if this is really a tendency or maybe only an artifact of the measurements. So that was a really surprising finding, and I think you agree with me when I say this was a significant reduction, almost complete reduction of tumor growth. And one more aspect that I wanted to give you is what we see in these tumors. So we then regained the tumors from these animals. So we sacrificed the animals and still tried to get the tumor mass out in order to do histology on these samples. And what you can see in these histological sections, this is mostly single cells and disseminated material, so it's not much what you can call a tissue, but we cut it out a little bit bigger so that we have a type of a tissue, a block, which we can use for histology. So what is important in this picture here is, with the wild-type virus, we still see some viruses, so there is still virus replication going on in the residual tumor. And we see the same, we see chimeric virus, but we see significantly more viral particles. So it looks like these viruses are pretty long, survive pretty long in this area. And when we look at fibrosis of tumor mass, so replacement of tumor mass by fibrosis, which is not a functional tissue, but it's a connective tissue, and it is a healthy tissue. And this brownish material indicates fibrosis, and we do not see any fibrosis in the wild-type treated animals, but we see significant and really convincing fibrosis in the after treatment of animals, of tumors with adenovirus, with chimeric adenoviral particles. Yeah, with this, I want to summarize my talk, and I hope I could convince you that this way to use sweet tooth viruses may be a strategy to treat tumors as long as they have the properties that we are searching for. But what is important again to mention is that almost all infections, viral, bacterial, pathogen infections, start with a contact made between the intruders and the host glycocalyx. This is by almost 100%, if not by 100% true. So this first contact may be a good source to find new specificities, new targets with which we can hunt behind tumor cells. So we used, in our case, we knew that polysialic acid is a factor expressed or an antigen expressed by tumor cells. So we made use of this knowledge to design these chimeric viruses to target cells, cover themselves with a polySia shell. And I could show you, hopefully, that these chimeric viruses were really selective and efficient in lysing polySia positive cells, not other cells. And we significantly, in the animal model, increased the lifetime or survival of these cells. Very important, we saw continued existence of chimeric viruses, and this is a very good sign because it demonstrates we have a continued anti-tumor surveillance in these animals. Or, yeah, at least we hope to have it. And these chimeric viruses reduce the cytotoxic effects that we normally see in oncolytic therapy. I haven't shown you any data of that, but this is a fact, and this is also because we have a much better, or one of the reasons why we have a much better fibrotic rebuilding of the tissue. So what is the future? We should find, based on this statement here, that we have many structures that may be unique in tumors, and we may have many intruders that have unique tools to identify these unique glycostructures. We should find more specific targets, and I know this is work going on also here in this institute, and we should, corresponding to this, identify reagents with which we can then selectively address these targets to destroy the tumor cells. Yes, with this, I'm at the end of my talk, and I have to acknowledge the people who really did the work. This is my PhD student, Nicolas Tim Martin. He has done all the experiments that I showed to you today and has been supervised by me and my good colleague, Florian Kühnel. Florian is an oncolytics person, and he was of big help and was amazing working together with him. So they are really highly talented young people. And here, this is my group at the Cellular Chemistry in Hanover. It's not very well to see, but these people are also in here. So, and with this, I should mention our work is funded by the Deutsche Forschungsgemeinschaft, and I appreciate this all, their help, their financial support, and I appreciate a lot your I thank you a lot for your attention and a lot for coming here and seeing this talk. Thank you very much.